Hello. Today we're going to look at cells and cell transport, transportation into the cell, the membrane structure, the methods for entry into the cell, and requirements for transport into the cell.
Good day. My name is Greg Johnson, and I'm going to talk to you today about the cell and getting things in and out of the cell, transporting things across the cell. The cell theory, or the cellular level of organization. Now, living things are made of cells. Sometimes a cell is all the living thing is. And essentially, they're the basic unit of structure and function, or of life, the basic unit of life.
Let's take a look now at the membrane, what it does, and what its structure is, and how its structure helps what it does.
We're looking today at cell movement, transport for the cell. Now, the cell's going to need things for its metabolism, for its stimulation, for its responses, for its metabolism. It's going to need these things. So they got to get in, right? But not everything gets in. Can't have everything coming in. So it needs to have a boundary. That boundary is what we call the cell membrane, right? It's composed of phospholipids. So the phospholipid makes up the cell membrane. It has a phosphate out here, which is charged. Phosphate is charged. And then it has fatty acids, or uncharged, nonpolar things, called lipid at the other end. It's got fats coming out of the other end. So the tails here are fats, fatty acids, and the big polar head here is a charged phosphate. Charged particles like to be with water. They're hydrophilic. Philiophilic, love water, hydro water. So they form this structure where the charged particle is where the water is. And since most everything, living thing is water or in water, there's water outside the cell and water inside the cell. Now, how do things get across? This boundary here doesn't let things that are polar through, not big things, very small things, yes, but doesn't let things that are polar charged through it. So it's selective, doesn't let everything through it, doesn't let large things through it. So how do things get in there? There are ways of getting them in. Proteins in the cell membrane, they act to bring things in.
Now let's go on to take a look at how these structures come together to create a semi-permeable membrane.
So this is what the fluid mosaic model of the plasma membrane looks like. It's basically, a good way to describe it is, it's like a great big sea of these phospholipids. So like an ocean, they're like the water in the ocean, all right? And these proteins are like icebergs floating around in the ocean, or ships, if you will. But they float around in there. Now here we have cholesterol too. Cholesterol helps make the membrane more movable so that the proteins can slide through it easier. So these are basically the components. These are basically the components of the membrane. Three components of the membrane. The lipid component, phospholipids. The protein component, molecules that float around like icebergs in a sea. The cholesterol helps the fluidity or the flowing nature of the membrane. All these things come together to create that barrier for the cell.
So what is it about these components that create a semi-permeable membrane across at the border of the cell? Let's take a look at how these components and the permeability of the cell.
Permeability of the plasma membrane. Water can move across the plasma membrane. So very small things, small molecules. Water is pretty small. Oxygen is small. Carbon dioxide is small. Small molecules can just drift across. They can get across. And even though it might be polar, it's not charged. So it can still get across. Specialized proteins turn aquapore and speed up water transport across the membrane. Poring, we'll look at that in a minute. Like a pore, it's a hole. Lets things through. Okay? Movement of ions and polar molecules across the membrane is often assisted by carrier proteins. And we'll look at how that works, but they can either carry it across or just make a hole to let it through. Make a hole. Some molecules must move against their concentration gradient using energy. We're not looking at that today. We're just looking at what doesn't use energy. Water outside the cell. Water inside the cell. So the hydrophobic phosphate is sticking out. Hydrophobic core. In this green channel here, you can see the hole right through the center. You see a channel or pore. There is charged molecules and ions. Charged molecules don't make it through. All right? Water will make it through because it's small and not charged, but water will also help through porins. They're called aquaporins. These help the water to get through it.
So we're seeing how proteins can help diffuse things across the membrane, or they can get across by simple diffusion. So either channel proteins help or simple diffusion.
Yeah, small molecules can get through. Macromolecules, very large structures, large molecules can't get through. All right, so this is just a look at what gets through. Some things can come right across the membrane. Things that are hydrophobic, like the tails, they'll let them through. Things that are large can't get through. There's a channel protein. So that's what the porins are, channel proteins. They allow passage of molecules through the membrane, being a channel in the protein.
So we've seen how channels work. Let's look at another protein method that uses a carrier protein to bring things inside the cell, transportation across
And
the
there
cell.
are carrier proteins combined with the substance to be transported, assist passage of molecules through the membrane. So what a carrier protein does is, it's on one side of the membrane, it'll just grab it, change its conformation, changes its shape, and when it changes its shape, it pulls it into the cell, let go of it inside the cell, then changes back to the shape it was in, to grab another one and pull it in. So these are how proteins work as enzymes. They generally change their shape over and over again to do their job, and then change back to the shape they started, so they can do their job again. But channel proteins, a pore or a hole, essentially, but it's a specialized hole. It only lets through specific things, not very many, not anything. Carrier.
Let's take a closer look now at how these proteins work. Let's look at their structure in the cell membrane and get a better understanding of just how they do their job.
Let's look at this. Channel protein allows a particular molecule or ion. It can even be an ion. All right, so particular ion molecule or even an ion, a charged particle. Charged particles can get through. They need something to help them across the plasma membrane into the cytoplasm. Carrier protein selectively interacts with a specific molecule or ion. Proteins always have their very specific thing that they interact with, so that it can cross the plasma membrane. You see here that it's actually grabbed hold of it, okay, and it's pulling it across. Changes its shape so that it pulls it across into the cell.
Now let's look a little bit at diffusion and how that helps drive this process of passive transport. And what is passive transport?
So just looking at this for a minute, here's a dye in a goldfish bowl. You drop the dye in, here it's a solid. The solid starts to come off as the water molecules interact with it. The water molecules dissolve it. And over time, it diffuses more and more into the bowl.
Diffusion is what drives the particles across the fishbowl via a concentration gradient. This same concentration gradient drives diffusion through passive transport, even using proteins that aid the process without energy.
You have to have that situation if you're not going to use energy. If you're working against a gradient, you need energy. The cell will use energy to do that. But passive transport doesn't use energy, it follows a gradient. In conclusion, we look at ways to get across the cell: diffusion, porins for proteins, and carrier proteins for proteins. The porins and carriers are specific to what they let in. The diffusion is specific to the size, basically, or the charge. All right. Thank you.